Hello students, welcome to the biology class. Today we are going to continue with our chapter biomolecules, and our today's topic is enzyme and its factors. Factors affecting the enzyme action are temperature, pH, substrate concentration and chemicals. So first we are going to discuss about the temperature. Effect of temperature. Enzymes generally function at the narrow range of temperature. The temperature at which the enzymes show its maximum activity is called its optimum temperature, means the most favorable temperature at which the activity of enzyme is maximum. Both below and above the optimum temperature, the enzyme activity decreases. At low temperature, the enzyme is inactive, while at the high temperature, the enzyme protein is denatured and so the activity is less. So what does it mean? For example, we keep our things in the refrigerator to have a longer life, because in the refrigerator the temperature is lower than the optimum temperature and the action of enzyme is inactive, means that becomes slow, and the growth of the bacteria, the growth of the another substances which can spoil your vegetables or the food product in that particular content, the activity of enzyme is inactive because of low temperature. On the other hand, at the high temperature, when we keep the things boiling at the high temperature, for example, boiling of the milk, also keep them favorable for the longer time, because the enzyme, which is protein in nature, is denatured and the action is less. So the activity will increase at the optimum temperature, that is maximum at the optimum temperature. But if the optimum temperature is lower than the optimum temperature, the activity will decrease or the enzyme become inactive. Similarly, when the temperature is higher than the optimum temperature, then the enzyme activity is going to be denatured, means again they become less active. For example, the optimum temperature is 35 degree to 40 degree. But if you keep the things at lower than 35 degree, means at the 10, at the 5, activity is decreased. The action of enzyme will decrease. Similarly, more than that, means at the 70 to 100 degree, again the action of the enzyme is going to be decreased, and the maximum action of enzyme is at the optimum temperature. So in this graph you can see the activity of the enzyme is there the maximum, because this is considered to be the optimum temperature. This is lesser than the optimum temperature, this is greater than the optimum temperature, but that is the most favorable temperature. The next factor is the effect of pH. You know that we are having a scale of pH from 0 to 14, and from 0 to 6.9, that is acidic, and from 7.1 to 14, that is alkaline medium, and the 7th is neutral. Enzyme, they are very specific, they are very particular for the pH. Each enzyme has particular optimum pH at which the enzyme activity is maximum. The changes in the pH of the medium alter the tertiary structure of enzyme, because that is of protein nature, and thereby decreases the enzyme activity. Again, I will give you one example. For example, we are having the enzyme pepsin. They are very active in the acidic medium, that is pH should be 2. Similarly, the enzyme pancreatic amylase, they are active at the pH 8.1, that is in the alkaline. If you alter the pH, the action is going to be stopped. So the enzyme, they are active only at the particular pH that is known as your optimum pH. So this graph again indicates this peak as an optimum pH in which the action is maximum. For example, it may be of a pepsin when its pH is 2, or that may be of your pancreatic amylase when the pH is 8.1, and so on. Next is the effect of substrate concentration. So substrates are the reactants which will attach with the enzyme, and as we have already discussed, the enzymes, they are having some specific sites known as the active sites. So with increase in the substrate concentration, the velocity of enzyme catalyzed reaction is going to be increased, but the reaction reaches the maximum velocity, that is V maximum, which is not exceeded by any further rise in concentration of substrate. Why? Because all the sites of enzymes, that is known as the active sites, they are being occupied by the substrate, means that condition has become saturated, and after that there is no free enzyme on which the substrate can bind, or they can make the enzyme substrate complex. So that means that is stable or saturated. This is because all the active sites of enzymes are occupied by substrate molecules, that is the enzyme molecules, and after the saturation there is no free enzyme molecule to bind to the substrate. The concentration of substrate at which half the maximum velocity of the enzyme reaction is attained is called Km value or Michael's constant. So in this graph you can see that as the substrate increases, enzyme increases, they goes on increasing because they are having a free active site. Once the active sites of enzyme, they get saturated, they become stable, that is called as the V maximum, and in between them when it is at the peak, and again it can increase a little bit, that is called as a half the maximum velocity. Next, a very important is effect of chemicals. The activity of enzyme is also sensitive to certain chemicals which bind to the enzyme. When the binding of chemical reduces or shut off the enzyme activity, the chemical is called inhibitors. So enzyme action can be inhibited in 4 different ways. Number 1 is competitive inhibition. Number 2 is non-competitive inhibition. Number 3 is allostatic and feedback inhibition. First, we are taking the competitive. As the name indicates, means the competition between the inhibitor, or you can say the substrate. So when inhibitor closely resembles the substrate in its molecular structure, because of the closely resemblance between the molecular structure, means at the same active site of the enzyme, both can bind themselves. So if instead of substrate, inhibitor gets bind to the active site of enzyme, the action is going to be stopped, and this is called as a competitive inhibitor. Malonate, example is malonate resembles succinate in its structure because both are the carbon 4 compound, and inhibits the action of succinate dehydrogenase. Such competitive inhibitors, they are used in the control of bacterial pathogen. So whenever the bacteria, they attack our body, cause some diseases, so we used to give a medicines in the form of the competitive inhibitors. So they are also known as the sulfur drugs. Drugs means medicines, which are very good and very important for curing the various pathogens. So in this image you can see that was a normal in which enzyme and substrate, they bind with each other and they can form the product, and the substrate is there. So inhibitor, that is molecular same to that of the substrate. Instead of substrate, the inhibitor is bind and the reaction is going to be stopped. Non-competitive means they are not resembling with each other. As the name indicates, they are not resembling, no molecular resemblance is there, but as the reaction is going on, in between site, they can attach other than the active site and makes the reaction stop. So when inhibitor does not compete with substrate for active site, they attach themselves to the part of enzyme molecules other than the active site and make a stable inhibitor enzyme complex. Such substances are called as non-competitive inhibitors. For example, the most of the poisoners, they are the non-competitive inhibitors. The very common and very well known is cyanide, which inhibit the enzyme cytochrome oxidase, which is essential for the respiration. Therefore, even the small amount of cyanide may prove lethal. Lethal means which can cause the death by destroying the cytochrome oxidase permanently. So the non-competitive, they are various poisons when they attach with a normal reaction which is going on, and the reaction is going to be stopped, and they may be lethal. Lethal means fatal, lethal means which can cause the death of a person. So here you can see this is the enzyme with the active site. The substrate will bind to the active site, but the inhibitor is going to bind other than the active site to form enzyme inhibitor complex, and the no reaction will take place, and the metabolically the reaction is going to be stopped. The next is your allostatic inhibition. In this case, inhibitor attaches to the specific site called as allostatic site of enzyme and changes the shape of active site so that substrate molecule cannot fit into it, and the reaction is going to be inhibited. So this phenomena is called allostatic inhibition. So this is the active site and this is substrate. It has to be bind with this. But when at this allostatic site, this inhibitor will bind, so this particular site that becomes not suitable for the substrate bounding. So that's why the reaction is going to be inhibited by the allostatic inhibitor. Feedback inhibition. Enzyme activity is also inhibited, reduced by the product of the same enzyme reaction. Such phenomena is known as the feedback inhibition. For example, glucose 6-phosphate inhibits the action of hexokinase that catalyzes phosphorylation of glucose. So as you can see, that is our reaction is going on. That is with the help of first enzyme you will get an intermediate product, then intermediate. Suppose this is your glucose 6-phosphate. This glucose 6-phosphate is going to inhibit the action of your first enzyme so that the further reaction is going to be inhibited. So this is called as a feedback. As you came to know that this product is sufficiently present in our body, in our metabolism, not required. So they themselves will go and inhibit the reaction. This is called as the feedback mechanism or the feedback inhibition. So with this we finish our enzyme, in which we have discussed about the nature of enzyme. We have discussed about the mechanism of enzyme, that is lock and key mechanism. Nomenclature, various classes of enzymes we have discussed, and today we have discussed the factors affecting the enzyme, that is temperature, pH, substrate concentration, and our inhibitors, that is chemicals. With this we finish the enzymes as well as the chapter biomolecule, which is very important. So please consult your NCERT for the same. Thank you and have a nice day.